Vamos ouvir a sua comunicação, que tem como título Perturbações do Espectro do Autismo, das moléculas a terapêuticas potenciais. Thank you.
Obrigado. Bom dia. I'll switch to English now for the sake of the non-foreigners in the audience. First I want to say I have a penchant for getting myself into trouble. From everything that I've heard and seen here at the Fundação Champalimaud, the doctors in the clinic are treating everyone appropriately. So there's no cause for alarm. It's all the other doctors that are taking shortcuts, because it's often not the experts at the highest level of their game, but the pediatricians, the boots on the ground, the general pediatricians in the community who are making these sorts of snap judgments, and it's not their fault because they have maybe 10, 15 minutes for each patient. So just to set that record straight, I'm going to talk about one approach to understanding ways in which we might find novel treatments for subtypes of autism. But I'm certainly not going to suggest that this is the only approach. I think that what you've heard from Trevor Robbins and other speakers studying social function in general, or the function of circuits and pathways, are equally, if perhaps not more important than the sorts of approach that we're taking. But as a neuroscientist by training, this is how we approach the problem, and in the process, hopefully we can gain a better understanding of how the brain works. And so when you hear about basic neuroscience that has no translational potential or isn't going straight to the clinics, that's what I'm talking about, and that's equally important as well. So we try to strike a balance, and it's all necessary. So as you all probably know, autism is not one disorder, probably. It's a heterogeneous group of disorders that we lump under one category, and that's based on the behavior that patients exhibit. They exhibit abnormalities in social interactions, abnormalities with communication or language, except in the case of Asperger's, where this is relatively spared, and they have a propensity toward repetitive stereotype behaviors, also known and have restricted interests and sometimes OCD-like, or obsessive-compulsive-like behaviors. 1 in 68 children are affected, at least in the United States, even higher incidence in boys. There's maybe 4 million children just in the United States, and certainly many more worldwide. And it's more common than a lot of disorders like Down syndrome or childhood cancer that get a lot more press than autism until recently. And there's no truly selective drug treatments, right? So there are good behavioral therapies. There are drugs that help with attention. There are drugs that help with repetitive stereotype behaviors a little bit, perhaps OCD qualities. People are on SSRIs or like fluoxetine, and then risperidone and aripiprazole are used to treat aggressive behaviors. But there's nothing that's truly selective for autism, and you might argue that many of the treatments that we have that are good for a lot of diseases aren't selective. We use a lot of blood pressure medicines for migraine. We use a lot of seizure medicines for chronic neuropathic pain. So that's not necessarily a bad thing that they're not selective. But what we'd like to do is get a better understanding. Now, as you've heard, anxiety, hypotonia or clumsiness, abnormal responses to sensory stimuli, or a craving for different types of sensory stimuli, intellectual disability, and seizures are also part of autism, but they're not diagnostic for autism. So our brain, as I had hoped Antonio Damasio would tell us this morning, is enormously complex. There's 100 billion neurons, 50 to 100 trillion synaptic connections, and everything we think and do is mediated by this singular organ. And what's fascinating to me is that our DNA has everything that we need to make a brain, along with a kidney and a lot of other complex organs. But if you mutate a single gene, it's known that in some cases this can cause brain diseases like autism. In some cases, all by itself, without other environmental inputs, we think, but we can't be certain about that. But how this works is fascinating, because we have 20-some-odd thousand genes, and the fact that single genes can cause this complex set of higher-order cognitive dysfunction remains a mystery today. So what happens if you have two brothers or sisters with autism and they're twins? Well, if they're non-identical genetically, then about 10% of them, in 10% of the cases, the other sibling is likely to have autism. In genetically identical twins, it's more like 70 to 90%. So you've got huge heritability and huge genetic component to autism, one of the strongest of the complex neuropsychiatric disorders. And so my lab is interested in how a mutation in a single gene might lead to changes in the brain that cause autistic behavior. And our approach is simple. We take known mutations in humans that cause or are linked to or highly associated with autism, and we change that same gene in mice in as close to the way as possible, and we ask, how does that change autism-relevant behavior in a mouse? Now, that's laughable to many of you, perhaps, but I'll point out that even if mice were people and they did behave just like an autistic patient, even if we do this, we've learned absolutely zero. We've learned nothing when we make a mouse mutation and see that it changes autism-relevant behavior in a mouse model. And I say that as somebody who spends my life doing that. And why have we learned nothing? Because if we choose the gene correctly, we already know that it causes symptoms in humans and behavioral changes in humans. So we don't need to do this experiment. But the experiment that we can't do as well in humans is asking this question, what's wrong with the brain? And that's really key. And so we use a variety of techniques to ask what's wrong with the brain. And just when you change one gene in such a complex organ as the brain, you may get 100 different things that are different as a result. And then the question becomes, well, can we fix this brain problem? And when we fix it, does this particular brain problem fix autism-related behavior or even non-autism-related behavior in a mouse model? So in some people's view, they want to see an autistic mouse. In my view, I want to see a gene create changes in the brain that results in some behavioral readout that we can test in the laboratory. And I don't care if it's autism-related or looks like autism or not. We just want to know how these genetic mutations can alter behavior. And then we ask, is this treatment useful in patients with autism? And I'll show you just a few examples today of how we're getting there. And the hope is that as we study the brain problems at multiple levels, as a result of these mutations, this will lead to understanding of the molecules involved and ways to improve treatments for autism, or at least a subset of autism. So we're investigating multiple genetic animal models. We're focusing mostly on genes that are important for synaptic function in the brain, and I hope that by the end of my talk I'll convince you that understanding brain dysfunction in these models predicts new treatments for autism, or at least a subset. And I'll start with, I'm not going to give you a barrage of data, or at least what I would consider not a barrage of data, but I want to talk about 2 or 3 examples of our approach. And the first one is probably the one that we've gained the most traction because a lot of basic science was already known about the pathways involved. So there's this gene called P10. It doesn't really matter so much what it stands for. It's phosphatase intensin homolog on chromosome 10. Mutations were identified in a subset of patients with autism, particularly patients with autism that have macrocephaly, or just big heads, okay? 2 standard deviations above the normal head size. Okay, and these were in conserved locations of this gene. What does this gene do? It participates in the PI3 kinase P10-AKT tuberous sclerosis pathway, where certain drugs or chemicals in the brain can activate this pathway and lead to protein translation and cell growth. When you lose this P10 gene, you overactivate this pathway, and that leads to abnormal growth. There's already an FDA-approved drug called rapamycin that can block mTOR kinase, one of the downstream targets. And mTOR is just mammalian target of rapamycin, so it's actually named after the drug that targets it. And so in collaboration with Luis Parada's group at UT Southwestern, we made a mouse that has deletion of this particular gene in certain parts of the brain, and we measured their social interaction in an open arena. And basically, these are the normal mice, the controls, and these are their brothers or sisters. And these mice are treated with vehicle or placebo. And what you see is there's a significant reduction in the social interaction between those 2 mice, okay? When we give the drug rapamycin, which blocks this overactivation of mTOR, what we see is this decreased social interaction reverses back to normal, okay? And rapamycin doesn't appear to be the sort of life of the party pro-social drug for everyone, because if you look at the control group, when they get rapamycin, they get worse or decrease their social interaction. So we think this is a relatively selective action of rapamycin on this overactive pathway in the model. Now, contrary to what most of us think, we don't expect that we can change the brain structure once it's developed. But the fact is, in this autism model, some of the neurons in the brain that have the mutation are greatly enlarged compared to the normal case, okay? That's better illustrated here. This green sideways less than sign, or V shape here, is called the dentate gyrus of the hippocampus. This is the dentate gyrus on the autism model. There's not an increase in the number of neurons. There's just an increase in the size and complexity of those neurons. And then this is the dentate gyrus after about 4 weeks of treatment on the drug. So it's not a perfect resolution, but these major structural changes in the brain are resolved by 4 weeks of treatment with rapamycin. In addition, this particular mouse model happens to have spontaneous seizures or epilepsy. They have about 1 to 2 seizures a day at baseline, and then during treatment with rapamycin, you can see that they go to almost having 0 seizures compared to the group that was treated with vehicle. Indeed, any remaining seizures goes down, the duration is significantly reduced, okay? So in this model, and this is a unique case so far, we can treat the brain weight problem, so the brain does get bigger. These mice have macrocephaly just like the kids. We can treat the neuron changes in synapse number and neuron size. We treat the social interaction abnormalities. I didn't show you the data, but we have evidence that these mice are anxious, and it treats that. It treats the seizures. And so in a very short order, this was all published in 2006 and 2009, we have an FDA-approved drug that can be tested in children now. I'll repeat that. It's pretty amazing that we have an FDA-approved that can be tested in children now. And it's beyond me why we don't have the money yet to do this study, although we're working with several sites to write grants to do this test. Now, is rapamycin going to treat all of autism? I don't think so. Of course not, right? Because all of autism patients don't have this mutation. In fact, only about 0.5% or less of autism has this mutation. But just the concept, I think, that we can, find a known cause for even 0.25% of autism, understand this pathway that it's involved in, and I meant to point out here that this pathway is not something that my lab worked out. This is something that's been worked out by hundreds of labs over decades of work, working in cancer cell models and just cell culture, right? So it's not neuroscience that worked this out, it's basic neuroscience, biochemistry that worked this out. So it's not someone working in an autism clinic that made these types of discoveries that lead to our ability to use rapamycin as a drug for this form of autism. So it takes all kinds of research, not just clinical research, not just basic science research, not just translational research. We all build off of each other, and I think that's important. Now, some of the first genes discovered in autism, aside from the sort of Fragile X, tuberous sclerosis, things associated with syndromic autism, were bridges across synapses in the brain. And those are these highlighted molecules called neurexins and neuroligands. Neuroligands hang out of the postsynaptic cleft and bind to presynaptic neurexins. And we thought that these are very, very rare, much more rare than P10 mutations. We're talking about, a handful of patients with each of these types of mutations, much less than P10, we believe. And we started by making genetic animal models of these mutations or deletions in these genes in collaboration with Tom Sudoff at UT South when he was at UT Southwestern. And I remember when we started these studies, Tom had already knocked out neuroligands 1, 2, and 3, all 3 types, and the mice still lived and they had synaptic transmission. So he's a basic scientist interested in how presynaptic vesicles fuse and how neurotransmitter gets released in the synaptic cleft. And he said, yes, they're involved in autism and intellectual disability, but there's nothing wrong with these mice. They release neurotransmitter just fine. But we were able to show that mutations in these genes or deletions cause profound, and even one of them causes profound abnormalities in behavioral function in the mouse model, and that led to further studies of their synaptic function, and we've identified abnormalities there that we haven't yet been able to treat. So neurexins and neuroligands bridge the synapse, and then they bind intracellularly to a gene called SHANK3, which I'll talk about briefly at the end. But I want to focus attention now on a member of the neuroligin family that's probably less well implicated in autism, but provides another example of how we've made some traction in understanding what's wrong with this mutant mouse and how we can maybe fix some of the things. So what we knew going into this was that neuroligin 1 is at synapses that are excitatory in the brain, glutamatergic synapses in the brain, that when we overexpress neuroligin 1, it leads to increased excitatory synaptic function in cultured neurons. And Tom Sudoff's lab had published that there's a decrease in the NMDA receptor-mediated response. So as many of you probably know, an NMDA receptor is a subtype of glutamate receptor that's critical for learning and memory in the brain and synaptic plasticity. And so we reason that if there's decrease in NMDA receptor function and nothing wrong with inhibitory synaptic transmission, that these mice would have altered synaptic plasticity, or LTP, decreased learning and memory on certain tasks, and they might have autism-relevant behaviors due to the links. And so this is the boring part of the story. When we measured synaptic responses at baseline and then gave a tetanic stimulation at high frequency for a very brief period, you can see that normal wild-type mice have long-term potentiation or an increase in their synaptic function, whereas the knockouts have a decrease in their expression of LTP. So that wasn't too surprising because NMDA receptors are critical for this form of synaptic plasticity, and they're dysfunctional. Also, when we put these mice in a spatial water maze task and ask them to find a hidden platform underneath the water, they can get better at doing this over multiple days. But when you trick the mice at the end and remove the platform and ask them where they spend most of their time, the normal mice spend most of their time where the platform was in this quadrant, whereas, and that's statistically significant over the other quadrants, but the other mice swim around at chance, so they're not using a spatial memory strategy to solve this task, okay? So that was the boring part, but what we found in this mouse model was not social interaction abnormalities. They're perfectly normal on 3 or 4 different social interaction tasks, but we found that these were the first mice that we found a significant and almost a doubling of the time they spent doing grooming, self-grooming behavior. So just like cats, mice, and rats groom themselves, they start by licking their paws, and they clean their paws, and then they clean their whiskers, and then they clean the back of their head, and then they clean their body, and they go all the way to the end of the tail if you watch them. And so this is an ethologically relevant, highly stereotyped behavior that mice do over and over again, just like we do. We groom ourselves almost every day. And then these mice, for some reason, were doing this more than double the control mice. And so we thought, well, this hippocampus isn't usually thought of as something that modulates grooming behavior. So we looked in the striatum, Rui Costa's favorite part of the brain, I believe, at least last week. And we looked at the NMDA and AMPA-mediated glutamatergic transmission, and we saw this decrease in NMDA receptor-mediated responses in the striatum as well. And so rather than be sort of basic scientists and go after, well, gee, what's this happening and what's the mechanism and how does neuroligin 1 interact with what protein that changes NMDA receptor function? Is it phosphorylation? We looked and simply asked the treatment-oriented question, is this change in NMDA receptor function in any way related to this change in grooming? And so to do that, we used a drug that's also FDA approved in humans, but probably won't work in this case in humans, a partial coagonist at the NMDA receptor called D-cycloserine. We used it at a dose that's known to boost NMDA receptor-dependent learning and memory. So these are the placebo-treated mice. Increased grooming in the green is the mutant. And then when we treat them with D-cycloserine immediately before testing their grooming behavior, or 30 minutes before, we see a complete amelioration of this increase in grooming. And you can see that the wild-type mice are not affected by this drug. Okay, so here's a molecular change or a functional change at synaptic connections that when we use a drug that rescues it, we can ameliorate a behavioral abnormality in the mouse model. And that's still a long way from treatment. But we wondered if maybe this is an off-target effect, maybe we're not affecting NMDA receptors. So we went to pharma, and they were developing glycine transport inhibitors. Glycine is necessary for the NMDA receptors to open. And it's tightly controlled at the synapse, so if you block its uptake with a glycine transport inhibitor, again, here's the vehicle dosed animals. These are the mutants up here, and then with successive doses of this sort of lead compound from pharmaceutical company AstraZeneca, it also ameliorates it. So two different ways of affecting or boosting NMDA receptor function can fix this problem in the mouse model. So this is the boring part of the story. Decreased NMDA responses leads to decreased learning and memory and synaptic plasticity. But more exciting to us is that something that may be relevant for autism, at least as much as it can be in a mouse model, can be alleviated by reversing the deficit in NMDA receptor function. Okay, so what are we doing now? We're trying to show that the striatum is the part of the brain involved, because this was a systemic administration of D-cycloserine, not into a specific brain region. So we want to know if we turn the gene back on in the striatum, is that going to rescue the behavior? If we attempt to rescue this by putting the drug directly in the striatum, will that help? And we're doing some detailed electrophysiology. Now we're getting into the nitty-gritty basic science, where we can now label different pathways through the striatum, both the direct and indirect pathways, and so that we know which neurons we're recording from, and we can ask, are there differences in the circuitry at a more detailed level? Those studies are ongoing. And I want to just finish up with one other quick example. I'm not going to show you a lot of data about SHANK3. We and others have made mice with SHANK3 mutations. What I can tell you is that SHANK3 mutations account for about 0.5% of autism. About 20,000 cases just in the US alone. SHANK3 is the causal gene in a syndrome called Phelan-McDermid syndrome, or 22q13 deletion syndrome. In that syndrome, these patients have autism, intellectual disability, absent or severely delayed speech, and about 75 to 80% of these kids have the formal diagnosis of autism using ADOS and ADIR, the research criteria diagnostic ways to show if a patient has autism or not. And in addition, patients that don't have this syndrome but have autism have been found with mutations in this gene at single points in the DNA, okay? And so SHANK3 is in the synapse, and it's a scaffolding molecule. It binds other molecules that link to metabotropic glutamate receptors, that link to NMDA receptors, that link to AMPA receptors. So it's mostly at excitatory synapses in the brain. And we're asking what I think is a critical question that I alluded to earlier. Do SHANK3 mutations cause autism because of irreversible neurodevelopmental problems, hardwired problems in the brain? Or is it possible that SHANK3 mutations cause functional changes in how synapses work, and then that causes autism that can be reversible in any way, shape, or form? And so the experiment that we've done is we've created a conditionally genetically reversible mouse that we can rescue after neurodevelopment is complete or anytime during neurodevelopment. And the way we did this is we took the normal SHANK3 gene. There are patients that have a mutation in exon 21, indicated by the star, where a single nucleotide is added to the DNA code and causes a frame shift and a premature stop codon in exon 21, okay? So presumably a truncated exon 21. And so we inserted this upstream of the normal exon 21, and just to prevent exon skipping, where we wouldn't get a mutation, we put a neo-stop cassette and then floxed the whole thing, flanked it with LOX-P sites, so that when we express and turn on Cre recombinase, we can revert back to the normal wild-type SHANK3 anytime during life. So it's an inducible reversal. So we cross this line to actin promoter driving estrogen receptor responsive Cre recombinase. We can administer tamoxifen to rescue the deletion or the mutation, and wait about 4 weeks and then study these animals. And so the goal, this is the preliminary data with wild-type heterozygous and knock-in mice. And you can see that the SHANK3 protein is gone in the knock-in mice with the mutation. And then after 4 weeks of the drug tamoxifen, the protein is back, so that we rescued the mouse successfully. This is wild-type heterozygous and homozygous mutants, and this is after tamoxifen. There's no significant difference in SHANK3 levels, and we've brought in the heterozygotes, and we've brought the complete deletion mice back up to heterozygous levels. So it's not a complete rescue, but it's as good as we can get. And we're going to look at behavior. We've actually looked at behavior and we're analyzing the data. We're looking at the changes in synaptic function and biochemistry, plasticity, EEG, ERP, MRI, and spine changes in the brain. So we've actually looked at multiple genetic models of SHANK3 mutations that have some relationship to hotspots and mutations in patients. So exons 4 through 9 we've targeted, exon 13 and exon 21. And when you look at the protein level in these different mutant mouse models, in the exon 4 through 9, you only lose the longest isoform of SHANK3. In this one, you lose the top two. and in these you lose all of them. And we've been comparing these serially in my lab and trying to find what's the same about these mutant mice, because many people have published on different mouse models of SHANK3 mutants, and they've identified differences in their synaptic function, at least where they look, usually in the hippocampus, and subtle differences in behavior. And so we're going through all these mouse models and saying, what's the common element across these models? And so far, the only molecular convergence we've seen is in the striatum, and we see a decrease in Homer1 BC. That's the long form of Homer. And we think that's going to correlate with a decrease in metabotropic glutamate receptor function and other things that bind to SHANK3 through Homer. But that's in common in all these mouse models. So the take-home message is genetic understanding. Genetic animal models are one part of the puzzle for understanding how autism is caused, what's wrong with the brain, how can we reverse these changes, can we treat this if we reverse these abnormalities? And so the combination of genetics from humans, animal models, and autism is one of the keys in the puzzle to helping autistic children. And so we've created what I like to call a comprehensive diagnostic and treatment center for mouse models of autism, which involves multiple forms of analysis. But what's missing here is collaborative clinical studies, which we're starting to get going, and also we're collaborating to look at patients with these specific genetic mutations, as I alluded to earlier, and are finding that we can see the same EEG changes in both the mice and the models. And thanks to my collaborators and funding sources. Thank you.
Thank you so much.